Proteins, the quintessential macromolecules, are essential to all processes sustaining life. The basic primary structure of proteins consists of a polypeptide chain of amino acids that fold into unique 3D secondary, tertiary, and quaternary structures that are intrinsically linked to the protein's biological function. Hence, understanding protein structures is a key step in unraveling the complexities of biological systems, advancing medical science, and catalyzing biotechnology. Due to the similarities between natural language and protein sequences, deep learning methods such as protein language models have revolutionized our ability to understand protein structures. Unlike older and much slower techniques like X-ray crystallography, these deep learning methods can quickly predict protein structure just from their sequences, helping scientists make discoveries without needing related previously known proteins. Deep learning methods can be used to determine the 3-state and 8-state secondary structure classes, which classify regions of a protein in terms of the characteristics of localized alpha helix, beta sheet, and coil formations. Additional capture proteins are protein backbone angles and numerical 1D structure properties such as half-sphere exposure and accessible surface area. These give insight to how the protein structure folds in its 3D conformation. Our training set consists of roughly 40,000 proteins submitted to the Protein Databank before 2016. Each protein in the input dataset is represented as a sequence of characters correlating to its constituent amino acids. We utilize mean absolute error, Pearson's correlation coefficient, and accuracy to evaluate our model outputs. Our baseline is the SPOT1D LM architecture, which incorporates one hot-encoded protein sequence inputs with embeddings from ESM-1b and ProteinBERT. It incorporates a 2-layer bi-LSTM with multi-scale resnets and a multi-scale res-LSTM from SPOT1D single, which is the previous iteration.
To adapt our selected baseline implementation, we propose leveraging the contextual embeddings from ProteinBERT as an input to the SPOT1D LM architecture. ProteinBERT is a deep language model that outperforms existing protein language models in terms of speed and size. ProteinBERT is a denoising autoencoder consisting of 2 parallel paths that distinctly process the local amino acid inputs and additional contextual protein annotation inputs. The model consists of 6 transformer blocks with 4 global attention heads per block and skip connections between hidden layers.
Our pipeline trains SPOT1D single distinctly for classification and regression tasks, creating task-specific ensembles. The model uses cross-entropy loss for the secondary state classification tasks and L1 loss for regression. For classification, SPOT1D LM averages the probability outputs from each model, while the regression takes the mean ensemble for ASA and HSE and the median ensemble for backbone angles.
This table shows the most significant results during our ablation study. We focused on these 2 main tasks, post-masking and pre-masking of the partially labeled amino acids. For post-masking, masking occurs during loss calculations, while for pre-masking, masking occurs during data pre-processing. We consistently encountered overfitting across our ablation study, where our validation loss started increasing at very low epochs. We see here that our post-masking validation accuracy was significantly lower than pre-masking. We also began investigating varying the composition of the ensemble to reduce overfitting. In our last study, we hypothesized that removing the LSTM layers will reduce overfitting. This is shown in the increase in number of epochs till convergence. We further investigated the accuracy of our SS8 prediction per the 20 standard amino acids for some of our test set. We see here that although our model has competing results with the baseline, it is not as generalizable across all amino acids. For the test states with proteins with known structures, our extended model shows competing results with the baseline model and only significantly falls short for amino acid D. Based on the results of our preliminary ablations, we have identified the following challenges: overfitting, limited data due to computational costs, and slow training due to small batch size. We plan to explore regularization techniques such as max pooling, dropout, and batch normalization to address these overfitting issues. We will also run training for our regression model to obtain prediction for protein backbone angles, ASAA, and HSE.